Anchored phosphatases modulate glucose homeostasis. We have chosen this particular paper because it is very recent. It's been published on the 31st of August by the EMBO journal, and it is relevant to glucose homeostasis because it looks at how anchored phosphatases such as aka P150 modulate glucose homeostasis. In this paper, a group of scientists from the US and Sweden have focused on revealing for the first time that AKAPs, which stands for alpha kinase ankyrin proteins, which are a group of multivalent binding proteins and are classified on the basis of their ability to interact with cyclic AMP dependent protein kinases, which is pKa. How those AKAPs influence the levels of insulin secretion and thus also the levels of glucose in the body. They do that by coordinating the spatial positioning of phosphatases, which are naturally occurring enzymes that counteract the effects of kinase enzymes. Therefore, AKAPs act as control points in the glucose utilizing mechanism. Insulin is released when glucose is present. So when glucose is present in the body, pancreatic beta cells release insulin via exocytosis. The process of stimulus, glucose being present, and secretion of insulin is a multi-step process. First of all, calcium 2+ ions influx from the cytoplasm into the beta cells and trigger fusion of insulin granules with the cell membrane. This process is modulated by cyclic AMP, calmodium, phospholipid dependent protein kinases. The actual release of insulin from the cell is triggered by several classes of protein phosphatases, including PP2B, which is a calcium 2+ dependent enzyme. The exact same group of protein phosphatases that are responsible for the release of insulin from the beta cells is also responsible for glucose uptake and utilization in insulin target tissues, which are skeletal muscle. This is where AKAPs play a major role because it is them that target protein kinases and phosphatases to cellular microenvironments where particular substrates, in this case glucose, are in abundance, and then AKAPs segregate their binding partners, substrates, to prevent disorderly change of signals. The methods of investigation that were used in this study include imaging techniques, immunoprecipitation, Western blotting, tissue analysis, as well as genetic modification of isolated insulin-secreting cells, and whole mice to investigate, obviously, the impact of anchoring proteins on glucose metabolism and insulin release. Also, hormone secretion studies, in vivo metabolism studies, isolated islet studies, and statistical analysis and molecular modeling took place. What is immunoprecipitation? It is a technique of precipitating a protein antigen out of solution using an antibody that specifically binds to this protein antigen. This process is useful to isolate and concentrate a particular protein from the sample containing many thousands of different proteins. So for example, on figure C here we can see an immunoprecipitation of aka P150 and aka P150D36 complexes. It also mentions co-immunoprecipitation of pKa2R2 and pp2b. Co-immunoprecipitation is basically immunoprecipitation, but it doesn't isolate proteins, but it isolates protein complexes. Also, as you can see, an immunoblot detection was made after the immunoprecipitation. Immunoblot is the same as a Western blot, and it is a widely accepted analytical technique which is used to detect specific proteins in the given sample of tissue homogenate or extract. It generally uses gel electrophoresis to separate native proteins by 3D structure or denatured proteins by the length of the polypeptide. The proteins are then transferred to a membrane, typically nitrocellulose or PVDF, where they are stained with antibodies specific to the target protein. Figure B here shows also an immunoblot of AKAP from the brain and isolated islets of Langerhans lysates. Lysates meaning preparations containing the products of lysis of cells.
Manipulating the AKP150 gene has metabolic implications for glucose homeostasis. The results showed that AKP150 normal mice secrete less insulin from B cells, yet display improved glucose handling because of increased insulin sensitivity in target tissues. These metabolically advantageous characteristics are also retained in mice that lack a 7 amino acid sequence responsible for PP2B formation. PP2B is calcium 2 plus ion dependent protein phosphate. Therefore, anchoring of PP2B is molecular and metabolic determinant that contributes to glucose homeostasis. This study has produced an enormous amount of data, and it is physically impossible to go over it in 8 minutes. Therefore, we have chosen a few first figures to show to you. As we can see in result A, several R2 binding proteins were detected in insulinoma cells, and it's been done by oral assay. And also we can see in result B that these included AKP150 and AKP220, two channel-associated AKPs that integrate calcium 2 plus ion and cyclic AMP signals. Gene silencing screen evaluated the role of each AKP in hormone secretion from INS1 cells and co-transfected human growth hormone known as AGH served as a marker of exocytosis. And under basal conditions, insulin secretion was unchanged from AKP150 depleted cells. In conclusion, we can say that the release of insulin is the main way in which the level of glucose are controlled in the body. That is why it's so important to study this area in more detail. If it is possible to develop drugs that target the region where anchoring proteins specifically interact with phosphate enzymes, it is feasible that insulin sensitivity could be improved in selected tissues, for example, skeletal muscle. Also, this would represent a valuable new molecular control point that might offer clinical benefits. Firstly, for diabetics. Secondly, individuals with other metabolic disorders. And lastly, all patients who are being treated with immunosuppressive drugs following